So welcome to the genetics PowerPoint. Like always, here's our objectives and our vocab. Okay, so you're going to be able to explain a genotype and a phenotype, tell if the genotype is homozygous or heterozygous, explain how a genotype is going to determine the phenotype for a dominant and recessive gene. You should know how to do a ratio, percentage, and probability using a Punnett square at the end, and you should be able to do a complete dominance Punnett square. Okay, here's all our vocab. That should all be on Quizlet. If it's not, let me know. So next, what's genetics? Genetics is the study of heredity. All right, this is going to be how characteristics are passed from one generation to the next, so from your parents to you and from you to your children. And it was found by Gregor Mendel in the mid-1800s, and we've done a lot of work on it since. So Mendel came up with 3 laws. The first one is the law of dominance. This just tells us that some alleles are dominant over other alleles. Then you have the law of segregation, which tells us that the genes are going to separate from each other when you make a gamete or a sex cell. Make sure you write sex cell next to gamete so you know what it means. And then the law of independent assortment, which says that genes for different traits are going to assort or separate independently from each other. So what that means is like your eye color and your hair color are not going to be based on each other. So first, the law of dominance. Some alleles are dominant and some alleles are recessive. Okay, dominant alleles usually produce a protein and recessives don't. That's why dominant alleles show and recessives don't, because they make a protein, and a protein is what you actually physically see. The next is law of segregation. So 2 members of a gene pair are going to separate from each other when sex cells are made. So if we look at this picture here, which is kind of crazy, right? These genes are not going to always stay together. In the end, you'll have big A. Let me just get my pen out so I can show you, or actually a highlighter. So you'll have big A and little a, right? But they don't stay together. No, sorry, big A and let's look at B. I don't want to look at little a. Big A and little b are not always going to stay together. Here's our big A's. Here's our little b's. In the end, we have big A, but it's not with little b, right? And over this side, we have some big A's with a little b, but they don't have to be together. And then law of independent assortment, right, is that the same thing, they're not going to stay together again. So genes for different traits assort independently when you make gametes, right? You can have black puppies, you can have yellow puppies, you can have puppies with curly hair, puppies with straight hair, it doesn't matter. So, Punnett squares. Punnett squares are tools used to determine possible genotypes of offspring, all right? You've done these before, I'm pretty sure. The probability of an offspring being produced are in the boxes. Each box represents 25%. I want you to think of the 4 quarters for a dollar, okay? 1 quarter is 25, 2 quarters is 50 cents, 3 quarters 75 cents, and 4 quarters you have a whole dollar. The parents are always going to go on the top. So when we do this, let me get my trusty pen out. Parents go up here. That's one parent. That's another parent, all right? And then the offspring, the babies, are these guys, right? The offspring go in the boxes. They're a combination of 1 allele from the mother and 1 allele from the father. The dominant trait is always going to show when we do complete dominance. And Mendel's first experiment is this one we have here. Okay, so what he did is he crossed a pure purple pea plant with a pure white pea plant. Okay, and the hybrids, so the mix right here, these guys are all going to show up purple because they have the dominant trait. So Mendel took 2 capital P, lowercase p offspring and crossed them. All right, that was what he did. Next, he took those babies and he crossed them. So we're going to fill that top allele. So these ones are going to go down and then fill the side ones over. And we always write the capital first. So here's our P's coming down. And the little P's are going to come down. Since they're little, they're going to go second. Then we have our P's coming over from the side and our little P's coming over from the side, right? So white comes back, even though the first time he crossed them, we all had purples, right? Now we have a white offspring. So let's say we have 2 parents that are heterozygous, right? We're going to complete the cross, okay? So heterozygous is 1 of each, right? We have 1 here and 1 there, okay? We know W is dominant, little w is, big W is dominant for white, little w is recessive for violet, Okay, so we bring in our top ones go down. So big W's, little W's. I put underlines into my little W's because it was hard to tell. My next ones go over. Big W's, little W's. Okay, and then I report my results. So you write your results from the cross by counting the squares. Okay, so if I do genotypes, this one is the only one that's big W, big W. Think of it as quarters of a dollar, that's 25 cents. This one and this one, right, are big W, little w, so that's 1/2 of them, that's 50%. And then this guy is little, little, he's my last one, he's 25% again. If you're counting the boxes, that's where you get your fractions. So 1 out of 4 boxes is WW, 2 out of 4 boxes is the heterozygous, one of each, and 1 out of 4 boxes is the too little. Okay, and my phenotypes is what they look like. So I'm going to do my phenotypes in purple, so that's a different color, right? This guy has a big W, this guy has a big W, and this guy has a big W. So all of those ones will be white because white is dominant. So I have 3/4 or 3/4, which would be 75 cents. So 75% or 3/4 is white. And my last one, 25% or 1/4 is violet, purple. You can also show the results as a ratio. So when you do a ratio, you just count the boxes. Don't make it harder than it is. And you always report homozygous dominant, heterozygous, homozygous recessive. So I have 1 homozygous dominant. I have 2 heterozygous, and I have 1 homozygous recessive. That's my 1:2:1 ratio. Looks like this. Okay, you can have zeros in a ratio if there's none. It's always homozygous dominant, heterozygous, homozygous recessive. All right, so now we're going to try it. And you should be writing these in the drawing side of your notebook. All right, so in pea plants, tall peas are dominant over short. So I'm going to write capital T equals tall, lowercase t equals short. Okay, and I'm going to make a pea plant for 2 heterozygous tall plants. So TC times TT. One of them goes on the side, the other goes on the other side. Make sure you keep the parents' alleles together. Oh, I read that wrong. My bad. It says it's crossing a, let me fix that, a heterozygous tall with a short pea plant. Sorry. Now my arrows are going to the wrong places. Oh my goodness. Let me fix this. This one goes there, and this one goes there. Better. All right, so top letter goes down. This one goes down into each block. This one goes down into each block. Those ones fly over. This one goes over. This one goes over. And make sure you put the dominant letter first. That's the way it works, right? And then I look at my results. What do I have? So for my genotypes, those are my letters. I have 50%, this one and this one. So 1/2, 2 out of 4 are going to be big T, little t. And I have 50%, this one and this one are little t, little t. Then I look at my genotypic ratio. So I count the boxes. I have 0 big T, big T. I have 2 big T, little t. And I have 2 little t, little t. Then I look at my phenotypic ratios or my phenotypic results. This is what it looks like. So let's remember before we even talk about this, big T is tall. So tall, tall, right? So I have 1/2 are tall or 50% tall, 1/2 is short or 50% short. And for phenotypes, I have 2 talls, right, to 2 shorts, 2 to 2. So in summary, when we do this, we're going to figure out the parents' alleles if they're not given. We make it Punnett square by putting parents on the top, 1 parent on the top, 1 on the side. Doesn't matter which goes where. I bring the alleles down, I bring them across to fill in my squares, then I list the possible genotypes, calculate the ratio, list the possible phenotypes, and calculate the ratio, right? It's homozygous dominant, heterozygous, homozygous recessive, or dominant to recessive.